work on this problem here. I'm going to grab my marker. All right, work on that question, please. This is a nice review from what we talked about last time, that we were starting to look at monohybrid Punnett squares. We're looking at one trait and the inheritance through complete dominance. So here we have two parents. We've got a homozygous dominant parent and a heterozygous parent. Let's set that up. Remember that we're looking at putting, if you have an individual who's capital P, each one of these represents a gamete, each P represents a gamete, and each one gets either a column or a row. So our homozygous dominant parent, capital P, each P gets a column. The other parent who is heterozygous, each of their P's gets a row. So big P, little p.
I have a question. The first two letters always go on the top, right?
Usually. Yeah,
Okay.
that's usually the way.
Or it's like the two capitals.
I mean, technically, it's always, if we're really getting technical, the male parent goes on the top and the female goes on the side. So, I mean, it doesn't, you're going to get the same outcome whichever way you put it. So remember, each of these represents, each letter represents a gamete, so each gets their own either column or row. We'll take this parent, we'll put them in the rows below. And we'll put the other gamete in the rows below. Looking at this other parent, the capital P goes in the rows across. I guess those are columns. And then this little p, the second allele from the parent, the other parent goes in the rows across. Remember that when you are calculating out the genotype and phenotype of the offspring, that your genotypic ratio always goes like this. Homozygous dominant, then heterozygous, then homozygous recessive. So now we take a look at our offspring and we count how many capital P do we have?
Two.
Good, we have two of those, right? One here and one here. How many big P, little p are heterozygous?
Two.
All right, we've got two. And how many homozygous recessive?
Zero.
Good. Okay, so our answer
5.
is E. I was like, do I have the answer up there? I do see it, right? Is E, that we have 2 to 2 to 0. Isn't this just looking? All right, 2, E is correct. Anybody, any questions?
What's the phenotype for that one?
Aha, okay. So let's say that I didn't give you this information. So let me give you that coded information now. So that if capital P, the allele, capital P represents the expression of the trait purple, and little p represents the expression of the trait white. The way that we would write out our phenotypic ratio is we would put the dominant allele first, the recessive allele trait second. So the dominant trait is purple. How many purple individuals do we have from our genotypic ratio?
6. 4.
Good. Okay, so the ones that are going to look purple are going to be homozygous dominant as well as heterozygous. So these two are always, when we're talking about complete dominance, the homozygous dominant and the heterozygous, as long as you have at least one capital P, you are going to look purple. And having two capital P's is not going to be more purple than a heterozygous. They are both going to show purple. It doesn't matter if you have two capital P's or just one, you are still going to look the same kind of purple. The only individuals who are going to show the recessive phenotype are going to be homozygous recessive. And we have 0. That's funny, I picked out a purple marker for this too. So our phenotypic ratio is going to be A, 4 purple and no white. Does anybody have any questions? So we've got another cross. All right, we're doing a little practice here. Okay, so now let's do a heterozygous parent with a homozygous recessive parent. So let's do this one. So go ahead and give that a shot. So this is the way we would set up this Punnett square. Let's take this parent allele for capital P, put them in the boxes straight down. Now, what I like to do when I have a little p is, just in case a capital letter comes along, I always like to put the recessives in the back in case a dominant comes along. Just this makes more sense to our brains, being this will mask that, rather than if you put, sometimes your brain may interpret this as, well, that little letter or the recessive masks the dominant, but that's not true, even though these are the same. So I stick that in the back. We have two little p's here. I'm going to put a little p and a little p across, and then again, a little p and a little p across. We are going to set our genotypic ratio by putting homozygous dominant first, then heterozygous, then homozygous recessive. How many homozygous dominant do we have for our offspring?
0.
Right, we don't have any. How many heterozygous? 2. So we've got one there and one there. 2 of those. And how many homozygous recessive?
2.
2. Got one there. So our ratio is E. It's going to be 0 to 2 to 2. Let's just say that we were going to pick our phenotypic ratio.
E.
Okay, yeah. Our phenotypic ratio for this one would be, in this case, these choices would be E. That remember that homozygous dominant and heterozygous are going to show the dominant phenotype. And homozygous recessive will show the recessive phenotype. And one more cross. So now for some of you, you're like, I got this, right? If both parents are only contributing the recessive alleles, the only thing that kids can get are recessive alleles. So let's take and just show that in our Punnett square. And again, what I like to do is when I have a recessive allele, just in case, I put it in the back. And here again, put that in the back. This parent here, now we're going to put a cross. Well, it's also a recessive allele, so no harm in putting it first. And then in our second row, so we are still going to put homozygous dominant, heterozygous, homozygous recessive. How many homozygous dominant?
0.
How many heterozygous?
0.
And how many homozygous recessive? Good. Let's just. So here, we have nobody showing the dominant trait. And all of our offspring will then be white. So 0 purple to 4 white. Anybody? Question? Mendel had a couple other laws. One we've already talked about, which is the law of segregation. This goes along with meiosis. But you get 2 alleles for every trait, right? 2 alleles for every trait, 2 alleles for every trait, 2 alleles for every trait. They separate during the formation of, if we're just talking about our regular cells, during the formation of regular cells. If we're talking about our gametes, you get 1 allele from each parent in your gametes. And then you get an N or haploid set from one parent and N or haploid set for the other parent. When you add those together, we start to get a 2N cell, which makes all the rest of our cells in our body. according to Mendel's law of segregation, an organism with the genotype big A, little a, will all the gametes have the gene big A? No, right? Will all the gametes have the gene little a? Half the gametes will have big A and half the gametes will have little a? I see some. Okay, let's read on. 3/4 of the gametes will have big A and 1/4 will have little a. I see some like, I don't know. 1/4 of the gametes will have a big A and 3/4 will have a little a. No, that's just weird stuff, right? So half the gametes will have a big A and half will have a small a, right? You basically are separating that capital A, little a into gametes. So half and half. Mendel also came up with the law of probability. The law of probability gives us an idea of the possible percentage of traits inherited by the offspring given our parents. So let's do this Punnett square. Both of our parents are heterozygous. Let's start at the top and we'll go in the columns down. Big T down. Here we have little t down. I'm going to go to the other parent and put a capital T across. And the last allele for the second parent, a little t across. So let's just say for coding purposes, big T is tall and little t is dwarf. We have one big T possibility. We have two heterozygous, big T, little t. And we have one little t, little t. So we have a genotypic ratio of 1 to 2 to 1. And then let's code this into tall versus short. Or dwarf, I said, sorry. Anybody with a capital letter, homozygous dominant and heterozygous, is going to look tall. And anybody with the two recessive alleles is going to look the recessive phenotype or dwarf. So we have a 3-to-1 ratio. Now, figuring out percentages, what you're going to do is you're going to divide each of these by the total number of possible offspring. 1, 2, 3, 4. So 3 divided by 4 is 75%. And 1 divided by 4 is 25%. So that's the way you would figure out the ratios, is that if you have 4 offspring, you divide each of the phenotypes by 4. So we have a ratio percentage of 75% tall to 25% dwarf. Based on the law of probability, what that means is that each child has this probability of traits that they inherit. So each child has a 75% chance of being tall and a 25% chance of being dwarf. It does not mean that all the offspring, whatever you have for offspring, that 75% of your offspring will be tall and 25% will be dwarf. Each child has that ratio probability. So inevitably, if each one has this like 75%, 25%, you could get all dwarf, if you have, like, let's say you have 3 offspring, and each one has this probability, and they just happen to all get that 25%, you could have 2 tall parents who have dwarf children. As the number of offspring increases in size, so like if you have 3, they could all be short, they could all be tall, it could be a mix. If you have, let's say, 100 offspring, your ratio will probably be closer to 75 to 25%. If you have 1,000, it will definitely be closer. Will it be exactly? No. So just to point out what this law of probability means. So again, each individual has a 75% chance of being tall and a 25% chance of being short or dwarf. All right, another practice problem. If the allele for inflated pods, capital I, is dominant to the allele for constricted, the cross big I, little I with little I is expected to produce what? So please do that problem. What I encourage you all to do is when you have a word problem like this, and it tells you what the alleles stand for, right away I encourage you to make a little chart like this, where instead of having to go back and read and find out what was the dominant allele for, just right away, right? Capital I means constricted, capital I means inflated, little I means constricted. That way you don't have to go back and read, because you know, test anxiety, you go back and we read things wrong. So I like to just like pull that information out, make this, so now I just look back at that and I don't have to go back and worry about, am I gonna read things incorrectly? So now I have the parents. I've got big I, little I with little I, little I. I'll make my Punnett square. Start with the parent on the top. I'll put the capital I in the rows going down. A column going down. I've got a little I. Because it's recessive, I like to put it in that back area. Now I'm going to go to this parent, little I across. And our second row, little I across. Now I'm going to calculate out my genotype and phenotype ratios. I don't have any big I, little I. I've got 2 big I, little I. And I've got 2 little I, little I. Translate that information into our phenotype ratio of 0 plus 2 gives me 2, and 2 little I give me 2 constricted. They want percentages, so I'm going to divide each of these by 1, 2, 3, 4. Oh. I can do math, I promise. 2 divided by 4, reduce that down to 1/2, which is 50%. And again, 2 divided by 4 is 1/2, is 50%. So now, when we take a look at our results, it says A, all our inflated pods are all inflated. No. We have some constricted, all with constricted, no. Half with inflated and half with constricted. Yeah, that one looks good, right? 3/4 and 1/4 and 3/4, 1/4. We don't have anything about 3/4 and 1/4. So that makes C our best answer. Now, some of you, I know, like, the thing about genetics is that when you get it, it's like, oh, I got it, right? When you don't get it, you're very frustrated. So some of you, I know, like walking into class last week, or last time on Tuesday, I started doing this, and you could just like look at what the 2 parents were, and you're like, I got all the answers. I figured it all out without even putting your pencil down. Some of us need to work out all of the information. If you need to like see just, you know, the way that I process through all of this, if you need to do that, do it. I always like to work these out. So some of you may have noticed that there is a way to do a shortcut here. Let's say that you have 2 pure breeding or true breeding parents. Remember, true breeding means 2 of the same. 2 of the same. Your gametes would then be, you just basically chop this in half, right? This is a gamete, capital T and capital T. For our homozygous recessive, a capital T or a capital T are your gametes. Now, normally, what we would do is we'd make a 4 square because you've got 2 gametes and 2 gametes. But when your gametes repeat themselves, so we've got a capital T, and then here's another capital T. This capital T will represent this one. So you don't have to show that. And when you have, again, a capital T or capital E, when they're the same, you only have to show one of each kind. So we could do a 4 square. So you could do your 4 square that looks like that. No, what am I, no, it doesn't look like that. Excuse me. You clearly. Sorry, I messed up. When you have 2 big T's on the side, and 2 little t's on top, all of the possibilities for the offspring are big T, little t. These parents can contribute little t's, these parents can contribute capital T's. So if we take a look at our ratio of offspring, given this, if we, let me green are our offspring. When we take a look at our offspring from our parents here, you have 0 possibility of capital T, capital T. We have 1, 2, 3, 4, big T, little t, and 0, little t, little t. Translate that into we have 0 dwarf, and we have 4 tall. Now, if we figure out our ratio percentage and we divide that 4 by 4 squares, 4 divided by 4, when we get a percentage, is 100%. Zero divided by 4 gives you 0%. If, let's say, we just did our shortcut here, and we only represented one of each kind of gamete, because when you have one that repeats, right, this row here, it's the same thing as that row. This column here is the same thing as that column. So when we do our genotypic ratio, we get 0 big T to 1 big T little t to 0 little t, which gives us 1 tall to 0 dwarf. When we do our reductions down, if we only have 1 square, we divide each of these 1 tall to 0 dwarf by 1, and we get the same phenotypic ratio. 100% to 0 and 100% to 0. So whether you do this the long way and you represent all of the gametes, or you only represent one of each kind of gamete, you still get all the same phenotypic ratio in the end. So if you're like, oh cool, I like this shortcut thing, do it. If you're like, I don't like that, that is a little confusing to me, do it the long way. It'll take you like an extra 3 seconds.
But you can only do it if the first 2 are capital letters and the last 2 are lowercase.
You can only do it if it repeats. So like, for example, on an individual who's this, you can't do a shortcut on this. Yeah, right.
Yeah.
Right, they have to be homozygous. Yes, correct. Yeah, they have to be the same.
Okay.
Good observation. Okay, anybody questions? So yeah, if you don't like the shortcut, if you're like, don't do it. Just like to point that out. Okay, next thing, we're gonna talk about test crosses. Test crosses is more of a word problem. With a test cross, you get like words. You don't get the actual alleles. It just tells you like this parent is homozygous dominant, this parent is heterozygous, but you don't get the actual genotypes of the parents. Test crosses work when one of the 2 parents is always going to be homozygous recessive. Then this will work out. So let's take a look. You have all this information already filled in? Okay, we can skip ahead to, all right, so let's skip ahead to this question. So can a purple pea plant crossed with a white pea plant produce a white pea plant? I'll show you the way I like to approach these. So, and it should say also, if this
We don't, yeah, we
the
don't have.
practical application part, I think you skipped the part.
No, she didn't skip. She thought it was filled out. And I told you no, but I don't think you had anything. These are before the slide, and this is the question.
Oh, wait. Cross the unknown.
It was the slide before, I think.
Oh, slide before, sorry,
Yeah, and
thank
you
you.
said it's filled out.
Okay.
This?
No, before. That.
Oh, sorry, thank you, yeah. Maybe the homozygous recessive part.
Yeah.
Okay, good. Okay, so now let's go to the example. Okay, so what I like to do here, and I also should add this, I always forget to do this in the notes, but if this were, let's say, an exam question, I would also say purple is dominant over white. So the way that I would approach this question is I would immediately code out purple, capital P is for purple and little p is for white. I'm going to pull out as much information from this word problem as possible. So I've got two parents. It says a purple pea plant. So we've got, I'm going to write our parents. One parent is purple and the other parent is white. And they produce a white pea plant. All right, so at least I kind of like mapped out what's going on. Now I'm going to try to figure out their genotypes. So my parent genotypes. If an individual is purple, what do I know in terms of them having two alleles for the trait of purple, right? Because I know there's two alleles for every trait, two alleles for every trait, two alleles for every trait. If they're purple, what do they at least have to have one of?
Big P.
Good. They have to have at least one big P. I know some of you have already got this. You're like, I got it. Let's hold off on this. The second one, if a parent is purple, that second allele could be a big P or it could be a little p, and they could still be purple. If my other parent is white, what two alleles do they have to have?
Little P, little P.
Yeah, they have to be little p, little p. So a white parent has to be little p, little p. It's the only thing they can be. So there's a couple ways that you can do this now. So if you want to do it the long way and see all of your data, you do both of these crosses. You do big P with little p, and big P, little p with little p, and see what you get. So if we take our big P possibility with little p, I'll put these big P's down, these big P's down, put little p across, and little p across. Do I get any white offspring? No, right? Okay, so just to show my work, I'll try the other possibility. I'm going to look at now the big P with the possible little p still being a purple parent with this white parent. Put the capital P down. Put the little p down. Put this little p across. And this little p across. Aha. When the parent is heterozygous, I know that we can get white offspring. The other way that you can do this is think about, okay, so we have a purple parent. This is kind of like using less data in this way, but using our data in a different way. That when I map this out, I can say, okay, the purple parent is big P something, and the white parent is little p, little p. The white offspring has to be little p, little p. Everybody has 2 alleles for every trait because those 2 alleles come from bio mom and bio dad. So, in order for this offspring to be little p, they have to get a little p from each parent. One of those little p's comes from bio mom, one comes from bio dad. So, if the purple parent has to have a big P, but also has to be able to contribute a little p, then I know that parent has to be heterozygous. And then the other parent is little p, so we know they're giving a little p. So that's kind of the other way that you can do it. Okay. Anybody, any questions?
Couple seconds. Says, is it the purple plant PP or PP?
The purple parent is big P, little p. They're heterozygous. Mm-hmm. So this would be our Punnett square that worked because we got the little p offspring. And if you just looked at it this way, our offspring are little p, and we know that you have 2 alleles for every trait because 1 allele comes from your biological mom, 1 from your biological dad. So this purple parent has to be carrying a little p in order to make the white offspring. And the other parent can only give little p's. Okay, so the question's answer is number 2, right? Big P, little p. One more practice question. So we are assuming that long eyelashes are dominant to short eyelashes. I don't know if this is true, I just made it up. A person with long eyelashes mates with a person with short eyelashes. The pair have 2 offspring with long eyelashes and 1 offspring with short eyelashes. What is the genotype of the parent with long eyelashes? Again, I know some of you are like, got it, don't do anything. You're very gifted. For me, I like to write everything out. So, for me, I'm going to write out all my information, and I'm going to write out long eyelashes dominant, short eyelashes recessive. So I'm going to do a capital L for long eyelashes and a little L for short eyelashes. And it tells me a person with long eyelashes. So I'm writing out my parents, a person with long eyelashes mates with a person with short eyelashes. And they have offspring. They have two, one with long eyelashes and one with short. So that's the way I would set that up. Now, I'm going to figure out what I at least know minimally for the parents. So the parent with the long eyelashes has to have a capital L with something else. And the parent with the short eyelashes has to be, yeah, two little L's. My offspring, long eyelashes, at least a capital L with something. And our short eyelash offspring has to be little l, little l. So you could do, as you have below, basically you want to do this Punnett square. But that turns into two different Punnett squares. The long eyelash parent, we'll try them with big L, and then we'll try them with big L, little l. And our other parent is little l, little l. So let's take a look at the homozygous dominant parent. Put big L down, and put big L down. Parent who is short eyelashes across, put the little L, and the little L, and again, little L, and little L across. And I see, I don't get any short eyelash children, right? So that doesn't work. So now let's take a look at our other possibility. I'm going to put big L in the columns down, and my little L in the column down. Take my little L's and put them across, and my other little L in the column across. Aha! You can get both long and short eyelashes. So this one works out better. We know that our parent has to be big L, little l. You also could have done it like I did the last one, second way. Okay, so you know that an offspring with long eyelashes is big L is something, but if they have a homozygous recessive offspring, we know one has to come from, one of the little L's has to come from mom, one has to come from dad. So this parent has to be carrying a little L, and the other parent can only give little L's. Anybody question? And so here's those same Punnett squares. Any questions about monohybrids?
Really quick, for this one that we just did, I was following along, but I still don't know how you got like you know where the question mark is?
Yeah.
Yeah, I don't.
To start with, right here.
No, the one where it's like above the box, next to the uppercase L, on the.
Oh, here?
Yeah.
So I'm just writing out the parents. The big L question mark,
Uh-huh.
that's one, that's this parent who has the long eyelashes. So I just made, at first I just mapped them out in a Punnett square, went big L question mark. And what I mean by that question mark is that that parent could be a big L or a little L and still be long eyelashes.
Okay.
So you could be these two genotypes, I should actually say, and be long eyelashes. But out of these two, I mean, you could work it out like this and show if the parent is this, with that short eyelash parent, this is the possible offspring. And if the parent is this, with the short eyelashes, this is the possibility of the offspring. So because this Punnett square shows both long eyelashes and short eyelashes for children, or the offspring, this
Would be correct.
question mark has to be
A lowercase.
a little L.
Okay. Gotcha. Makes sense.
Okay, good.
Thank you.
Anybody else? Did I tell you all there are 2 alleles for every trait? Did I tell you that? Okay, well, let's remember there are 2 alleles for every trait. 2 alleles for every trait. That is not what I want. I want. Okay. For 2. Okay. One more practice for monohybrids, and then we are going to move on to something else. What is the ratio of phenotypes in the offspring produced when a flower with the genotype big P, little p self-fertilizes? So they're acting as both parents, big P, big P, little p, times big P, little p. And we're assuming complete dominance for this trait. So go ahead and work that one out. So hopefully your Punnett square looked like this, where you put a big P, little p parent on the top and a big P, little p parent on the side. We ended up getting 1 big P, homozygous dominant, 2 big P, little p heterozygous, and 1 little p, homozygous recessive. Big P, and big P, little p are going to show the dominant trait, and little p is going to show the recessive trait. It wants us to figure out percentages, so I'm going to divide each of our phenotypic ratio numbers by 4, because we had 4 boxes up here. So 3 divided by 4, that's the worst score ever. Three divided by 4, or 3/4 is 75%, and 1 divided by 4, 1/4 or 25%. So 75%. So what this means is each of our offspring has a 75% chance of being dominant and a 25% chance of being recessive. So where's our 75-25?
C.
Good, C. So C is correct. Anybody, any question on that one? Yes.
This is a related to this, but when am I exam 3?
When is the exam?
Yeah.
The 22nd of April. I moved it to, it was supposed to be on the previous Thursday, but I don't, sometimes I'm like, I question my younger self. But I went, wait, why would I put it on a Thursday when I like to make it on a Tuesday so that you have the weekend? So I moved it to the following Tuesday. So it's on, the next exam is on Tuesday, April 22nd, to give you the weekend to study also. How many alleles are there for every trait?
1.
There's 2 alleles for every trait. 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 So, we are going to do what are called dihybrids. Di means 2. We're going to look at 2 traits at one time. So we're going to look at our parents, and we're going to look at how they pass on 2 traits to their offspring in one Punnett square. So tall, capital T, dwarf, little t, capital R, red, little r, little red.
White.
Sorry. You're right. Okay, so how do we get our gametes now? This just makes it a little bit more complicated to get the gametes. You know that term FOIL in math, first, outer, inner, last? We're going to use that. Okay, so we're going to call this a true breeding dominant parent. They're going to be dominant for both this trait and that trait. There's 2 alleles for every trait because we've got 2 T's and 2 R's. So the way we get all of the possible gamete combinations, because before it was just easy, right? If it's this, our gametes are big P or big P. Now, what do we do with that, right? Okay, so now we're going to use the FOIL method. So we're going to take our first letters. So FOIL, first. Take the first T and the first R. First, inner. The inner T and the inner R. Wait, let me move this down. Getting in my own way. First, inner. Now we're going to take the outer letters. The outer T and the outer R. And then we're going to take our last T and our last R. So take our first letters of each of our traits, our inner letters of each of our traits, our outer letters of each of our traits, and our last letters of each of our traits. So that's the way we would FOIL. It gives us all the possible combinations of these T's and these R's that this parent can give. Anyway, question on FOIL. Nice little review from some math somewhere. So our gametes for our parent, that was big T, big T, big R, big R. If we do, let's do little t, little r, little r. We've got them up there. So again, remember, the first T and the first R. Our inner T and our inner R. Each of these is giving big T, little r, big T, little r. Our outer T, and our outer R, little r. And then our last T and our last R. And we got all big T, little r, big T, little r, big T, little r, big T, little r. So now, in terms of our Punnett square, we have 4 gametes, right? Four gametes and 4 gametes from our foiling. So now we're going to do a 4 by 4 Punnett square. But before we do that, let's FOIL this one. For this parent, we're going to take our first letters, big S and big Y. We're going to take our inner letters, which would be little s and big Y. Going to take our first inner outer letters, which is big S and little y. And then our last of each, little s, little y. So we get 4 different possible gamete combinations. It's got to be E then. Anybody questions on foiling? Okay. You're going to have a 4 by 4, right? 4 gametes by 4 gametes, a 16 square Punnett square. Let's do it. Sorry, you didn't get that one. Okay, ready? Are you ready? Are you ready? So here's our Punnett square. We'll put big T, big R across. Just like our monohybrid, each of our gametes gets either a column or a row. So I put the homozygous dominant pure breeding parents' gametes on the top. I'm going to put our heterozygous pure breeding parents' gametes on the side, which were little t, little r. Okay, how do we keep this organized? So, kind of think about rules of thumb for this. You're going to have 4 letters in each box. 2 come from mom, 2 come from dad, or vice versa. You're going to have 2 T's and 2 R's. Keep your T's in the first 2 places, keep your R's in the second 2 places. So let's go with these straight down. The way I would do this is I would go big T, space for the next T, big R, and the first R space. I'm going to do that straight down. Big T space, big R space. Big T space, big R space, big T space, big R space. We have to do this for all 4 columns. Big T, big R. Big T space, big R space. 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 Hopefully you got that down. Be careful with your writing. Okay, so all of them. Big T, space, big R, space. Put them down, 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 down the columns. Our little t, little r going, the space is left open. So little t, little r across, little t, little r, little t, little r, little t, little r. And to the next row, little t, little r. This one's pretty straightforward, hopefully. I'm going to go through the offspring, and then we'll go to lab, and you'll get a break. So please hang in there. I highlighted the first box because if you like that shortcut idea, these are all 4 the same. So if you want to do the shortcut, you only have to represent one of each kind. You could get rid of these 3 other columns. And if you want to do the shortcut on this parent, you could get rid of these. And your Punnett square, if you want to do the shortcut, is that. Because we're going to take 1 out of 1, 100%, or 16 out of 16, 100%. They're all the same. All the offspring are 100% the same. So if you like the shortcut, do it. If you don't, you're like, no, 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 no. Okay, so this is in reporting our offspring. Remember that these are the F1 offspring from those parents. Those are the first generation of offspring, the F1 offspring. You could just do it easily. You could say all T, all capital T, capital R, little r. You could say if you did 16, and if you did the shortcut, you could say 1. We could do the same with the phenotypic ratio. They're all tall red flowers. You could say 16 tall red flowers. You could say 1 tall red flower. Now we're going to do a crossbreeding thing, and we're going to have 2 of these mate. So we have to foil to get our gametes. So foil, foil, foil. Okay, first letters first. Big T, big R. Inner, little t, big R. Outer, big T, little r. And our last, little t and little r. Those are our 4 possible gametes from each of our parents, because we're going to mate them together, or we're going to do a self-mating thing. Both parents are going to be big T, little r, little r. So parents, big T, little r, little r. And when we foil, we get those gametes. Now we're going to put our gametes on here. Okay, so now this gets a little more complicated. Let's start at the top and go in the columns down. Remember, keep your T's together, keep your R's together. So in this column, I'm going to put big T space, big R space, big T space, big R space. Big T space, big R space, big T space, big R space. The big T, little r. Big T, space for the next T. I'm going to put a space in case a big R comes. I'm going to put that little r in the back. So I'm going to do big T, space, space, little r in the back. Big T, space, space, little r in the back. Big T, space, space, little r in the back. Little t, big R. Space, little t, big R, space. So I'm putting this little t, big R in the middle of the 4 spots. So space, little t, big R, space. Space, little t, big R, space. Space, little t, big R, space. And our last column, I'm going to leave a space, put a little t, space, little r in the back. Space, little t, space, little r in the back. Space, little t, space, little r in the back. Space, little t, space, little r in the back. And now, we're ready to go across. So I've got big T, and I've got another big T, and another big R. So we're putting in a big T and a big R. Across, big T, big R, across, big T, big R, across, big T, big R, across. Our second row, we've got a big T and a little R. Big T, little R, big T, little R, big T, little R, big T, little R. Our third row, we've got a little t and a big R. Little t, big R. Little t, big R. Little t, big R. Little t, big R. And our last row, we've got a little t and a little r. So little t, little r. Little t, little r. Little t, little r. Little t, little r. Oh boy, there's a lot of different genotypes now, right? So, I like to do a rule of thumb. If there's more than 4 different genotypes, do not count out the genotype ratio. Just forget it, too much work, right? This. Okay, there's too many different genotypes, so forget it. Let's just try and figure out what the phenotype ratio is. So, the phenotype ratios. You've got tall red, you've got tall white, you've got short red and short white. Those are your 4 possible phenotypes that are in here. So that's what we're going to focus on. So based on so you have this tally mark below. I'm going to put it over here. Tall, red, tall. Let me put it over there. Okay, so, and then I'll show you how I like to tally. So we've got these 4 possible phenotype ratios. I always put the 2 dominant first, the 2 dominant last, the first dominant with the second recessive, and then the first recessive with the second dominant. That's the order they always go in. Okay, so you have this all stacked nicer in your notes. But what I like to do is just go box by box. Start here. What does this look like?
Tall red.
Tall red, cross it off. Tally it over here. I'm telling you, cross them off. Or make, if you're like, I don't want to cross things off, make a little notation that you counted that box, because it's inevitable. If you just kind of go box by box, and you miss one, and then you're like, oh crap, which one did I miss? Okay, so we'll go across. What's this? Tall red. Boop. Big T, little t, big R, big R. Tall red. Tally. Big T, little t, big R, small r. Tall red. Start in our second row. Big T, big R, little r is a tall red. I've got 5 now. I do the 4, and then across the 5 to tally. What's this one?
Tall red.
This one.
Tall
White. No, yeah. Tall, white. Yes. Tall, and white. This one. We're back to
Tall red.
tall red. And then this one.
Tall white.
Tall white. All right. Third row.
Tall red.
Tall red. Yeah, I know this gets a little delusional. Another big T, big R, little r, tall red.
Dwarf red.
Dwarf red. And dwarf red. All right, our last row, tall red. Tall white. Dwarf red. And one little dwarf white. Now I add them up. I've got 9 of these, and 3 of these, and 3 of these, and 1 of those. Whew. Our phenotypic ratio, when we have 2 parents, when both parents are heterozygous for both traits, you get a phenotypic ratio of 9 to 3 to 3 to 1. See how I put them in order, right? Both dominant, dominant recessive, recessive dominant, both recessive. 9 to 3 to 3 to 1 phenotypic ratio when both parents are heterozygous for both traits. So if you're doing a problem and you're like, oh wait, I'm supposed to get 9 to 3 to 3 to 1 and you get 8 to 4 to 3 to 1, something is awry. You've miscounted, okay? So those are your basics of the Punnett squares. Let me just make sure you got your notes. That's what we needed. So this is, for example, I have it colored in. You know, you could do this if you're more like to color. You could do colors on your lab like this instead of crossing off. You could color them and see the 9 to 3 to 3 to 1. So phenotype ratio of 2 parents heterozygous for both traits, 9 to 3 to 3 to 1, always. That's always going to be the case. Okay, let me see, is there anything else right now? And this is just another